This is Breakthroughs, a podcast from Northwestern University Feinberg School of Medicine. I'm Erin Spain, host of the show. Millions of molecular proteins are swimming through our body's cells, and many studies have discovered that these proteins are the main drivers of all human diseases. Scientists are now mapping proteins the way the Human Genome Project mapped genes. Northwestern's Neil Kelleher is at the forefront of the Human Proteoform Project and is here to explain how it could lead to more targeted and effective diagnostics and treatments for diseases, and how proteoform research at Feinberg is expanding. Welcome to the show, Dr. Kelleher.
Thanks, Erin. Great to be with you.
So explain exactly what a protein is and what the proteoform is and the critical roles that proteins play in our bodies.
So it turns out that the genes are the instructions for how to make proteins in our bodies. And if you just point to anywhere in your body, you'll be pointing to proteins. Your hair, your eyeballs, your bones. I mean, we are built up of proteins, and therefore it shouldn't surprise anybody that they are central to when we get disease. They're mediated by proteins. Their proteins are going wrong, especially in neurodegeneration. So like Alzheimer's, Parkinson's, and ALS or Lou Gehrig's disease. It's just, these are proteinopathies, people even call them. At the molecular level, proteins are strings of amino acids, and they can be extremely long. They can be 500 amino acids long, 1,000. Some of the biggest genes, like Titin, is a gene that's one of the biggest ones. But then what happens is they fold up and they get decorated. That's a protein molecule. And, a lot of people have heard about AlphaFold or Google's collaborative response to fold them up. But you cannot predict all the decorations. And that's the molecular decorations. That is what can turn an enzyme on or off, which an enzyme is a kind of protein. So these decorations are absolutely critical to understand how we work at the molecular level, at the protein level for human biology.
So you just published a paper in Science Advances with your co-authors defining the Human Proteoform Project. Tell me about that paper. Why is it so important?
The Science Advances paper that came out on November 12th, that was years in the making. And we at the Consortium for Topdown Proteomics, which is anchored by Northwestern, and our proteoform informatics team is top shelf. We have a lot of great, 60 people at Northwestern Proteomics. And we've, for years, anchored the consortium and brought more people in. And there is a crescendo happening, Erin. You're absolutely right. People all across sectors, government, private sector, academics especially, are now jumping on. And it's really exciting to see. And this paper was designed as a watershed moment. It's like, here would be the framework. And it outlines the case for, the proteoforms are involved in all human disease. The proteome is comprised of proteoforms. So let's get back to basics. And that proteins are a critical connection between the genome and our diseases. All human diseases involve proteoform biology. So therefore, proteoform measurement is the key linkage between our genome and our blueprints of life and then actually living life. And this will connect up what is absolutely required to get more precise about all our biology, which means better therapeutics, better process of creating drugs to intervene, more precise ways to intervene, also informed by precision genomics and regenerative medicine. All the goals for our century require this as a next obvious step. And that's what the paper was meant to do. And we've sort of kept our powder dry for some years, but this is the decade. There's a question, why now? And there's a lot of readiness across sectors. Like the private sector just put in almost $3 billion by some counts of investment for already next generation proteomics, single molecule, single cell proteomics. These things are moving aggressively. And so there is a role for a government-funded project to fill that the reference proteome will be available. And we can all work in parallel to accomplish what took 20 years in the Genome Project to do the project, and then disruptive genomics, next-gen sequencing called NGS. All of that was after the project was done in 2002. So it was like the Genome Project was done, $1 per base was reached in 1994. Then the project was done, shake hands with Bill Clinton in 2000, 2002 to 2008, immense gains through the private sector, 300,000 jobs a year created, just all this incredible things. And that's really what the proteome, the wild frontier, we got to domesticate it.
How do you analyze proteins and proteomics? What specialized equipment is needed?
So the proteoform is just that specific molecule. And how do we detect, analyze, characterize these decorations that are captured by this word proteoform? A proteoform captures all the sources of variation of a protein molecule, and it does so precisely. And how we measure those is currently with an approach called mass spectrometry. So we need to systematically discover and characterize proteoforms at about 100-fold less expense than it currently costs. So that's one expression of this project, the Human Proteoform Project. And currently, mass spectrometry is really the only way to assert and discover the proteoforms. What you do after that could unlock so many other possibilities that may or may not use mass spectrometry.
So you use something called top-down proteomics. Explain top-down proteomics versus bottom-up proteomics.
I like a stamp collector's metaphor. So if each proteoform is a stamp, and that's what's created in our bodies from the 20,300 human genes, and there's going to be 50, 100 million of them is our estimate. Okay, then we have to collect stamps in different cell types, in the blood. We have to really determine these molecules because that's our biology. It's what is the proteome, or the collection of all proteins is the proteome, like the collection of all genes is the genome. So the way that we do this is top-down proteomics. It's the direct, no inference required. And you first weigh the proteoform down to the last hydrogen atom. We can tell the molecular composition of that proteoform. And that's the top-down philosophy. So we first weigh it, and then we controllably fragment it into pieces, and we weigh all those pieces. That's top-down. And in a stamp analogy, it's equivalent to taking a picture of the stamp, and so you know the whole thing, and then you can characterize each little pixel on the stamp. That's top-down. Imagine now, here's what bottom-up proteomics is, which is a fantastic $4 or $5 billion a year market. It's how we know the low-resolution draft of the proteome. And it's how we've discovered so much about all the protein machines that drive our biology in proteomics. So what we're saying is we need a new ecosystem on top of bottom-up. So it's a proteoform resolved and proteoform enabled world that we envisage. And the way to do it, or the difference, is to complete the stamp metaphor. What we do in bottom-up proteomics is we take a collection of, say, 10,000 stamps from the blood. We draw blood, there's 10,000 proteoforms in there. Okay, what we do is we shred them into an average of 50 pieces. And then we take a giant fan onto the table with all those pieces, and we splay all the pieces out. We collect maybe 10%, 8 to 10% of the pieces of the stamps back, and we're trying to put them back together each, and its limitations are becoming clear. So the top down is every stamp is kept in its lane. Every proteoform is kept in its lane properly for characterization.
Is this more difficult to accomplish, the top-down proteomics?
Yeah, and there was one guy in 1999, I suppose, or one person. That was me. I started my lab with this idea, like, weigh every protein. And some people really liked it. Some thought, okay, well, he's in trouble for tenure. He might not make it. But it's in the tradition of American science to throw out really ambitious goals. Or in Chicago, too, like Burnham's Make No Small Plans.
This is becoming a much more accepted way to do science when it comes to proteomics. Is this the method that will be used in the Human Proteoform Project, the top-down proteomics?
Well, in the first phase of the project, yes. Anybody's crystal ball maybe goes 3, 4, 5 years to... And like in the Genome Project, there was disruptive elements at work, and that's what we want. We want cheaper, better, stronger to deliver value and understanding to all the stakeholders of the project. And the American people are some of those. So we want to imagine that we can do the project. And that would be first phase would be use top-down proteomics to assert proteoforms. Once they're discovered, though, and characterized, there's the atlas, the Human Proteoform Atlas. And then all sorts of other technologies can use that information, including bottom-up. Now I can devise assays knowing the decorations on the proteoforms. So everything becomes proteoform enabled. And then next generation proteomics, just like next generation sequencing, we can now sequence a genome for $1,000. It's absolutely incredible. So that's what we need in this decade for the proteome.
Here at Feinberg, you're doing some work in medical research with proteomics. You have collaborators in organ transplantation and cardiology, and you're finding ways to integrate proteomics into that research and clinical care. Tell me about some of those projects happening right now at Feinberg and the advances you've made.
Let me just make a point about the proteoform measurement impacting clinical care, because that in proteomics, if you go back to 2002, cancer detection early through blood biomarkers, right? That has proven to be very difficult. And the last 20 years has borne that out. But there's one area where actually proteoform measurement as the vehicle has already led to a clinically deployed assay in over 3,000 hospitals worldwide. And it's called the Biotyper assay. And it's the cheapest way to determine the bacterial pathogen in someone's infection. So if someone has a bacterial infection, there's a clear way. You look at a bunch of what's called ribosomal proteoforms, and those actually tell you what bacteria. So that proves the business and science case. That is often overlooked in our field. But other complex Mendelian disease, or, that's a fancy way, it is to say most complex disease involves multiple genes. It's highly complicated biology mediated by proteoforms. And that point of looking at complex disease is what we do every day at Northwestern Proteomics. And we have 4 disease areas that we work on: neurology, you mentioned cardiology, immunobiology, which is our transplant center, has been a fantastic partner. The CTC at Northwestern is just absolutely spectacular. So many collaborations there. And then neurodegeneration is the 4th area. So we show the value of proteoform measurement in basic and in clinical research. So patient cohorts, we have a fantastic collaboration with Josh Shalitsky and also Don Lloyd-Jones and John Wilkins in cardiology. And we have an initiative called the NPI, the Northwestern Proteomics Initiative, that's been supported by Eric Nielsen, the dean, and the upper administration. And that will expand the number of targets that we can look at to discover, and that the proteoforms, like, okay, I can give you one in oncology and KRAS. So KRAS is a gene, it's an oncogene, and it's mutated in so many cancers, but like 90% of pancreatic cancer. And we've mapped 37 proteoforms of KRAS, and we're showing which ones matter in the dynamics of the disease. And then KRAS is also a drug target. The only way to monitor how much the drug is binding to the target is through proteoform and measurement. And we've done that with collaboration with the National Cancer Institute, as well as, collaborators here at Northwestern. We have at any given time like 120 labs that we collaborate with at Northwestern Proteomics. It's about 60 people.
You wear a lot of hats here at Northwestern. You mentioned Northwestern Proteomics. You're also the director of the Chemistry of Life Processes Institute. Tell me about the different roles that you're playing right here at Northwestern, how they all kind of work together.
There's just a natural alignment there because both want to embrace Feinberg over the 10 or so miles that separate us geographically and do more with the investments that we have. Basic scientists on the Evanston campus need to understand unmet clinical need. And I'm among them. I absolutely, when I see a gap, I know what I have to build my tools for, whether they're diagnostic tools or chemical tools, synthesis of new drug molecules. I need to be on point and on target to compete for resources out in the ecosystem of funding for science. And that's what the function of the Institute is. And though my personal passion is the diagnostic side, using proteoforms as biomarkers, I have a whole set of chemists and engineers under the roof of CLP that have the same need. So the interaction with the School of Medicine is so critical.
So what can we expect over the next 5 years, 10 years from the Human Proteoform Project?
Getting support for the project in the US federal government is an obvious thing we're trying to do. Other funders around the world were able to come in on the Genome Project, like the UK's Wellcome Trust, hooking these things up and getting the project on the rails. And I think the next 5 years, we'll see the expansion of the proteoform discovery effort, dropping it into an atlas, putting that atlas in a form that people can absorb and start to use immediately. We don't have to wait 10 years, no. But also there's a clear script now that scientists run, which is much more collaborative, much more diverse and embracing inclusion. And that is through the consortium, we have a egalitarian way to make sure that progress is done aggressively, but in a process that listens to the community. And all those things can happen in the first 5 years. You go to the 5 to 10 year thing, that's when the private sector can really start to contribute. And unlike the Genome Project, I'm trying to have that disruption, trying to get the project going, but then try to embrace all those ideas that would really just help us get to the goal line. And there was, as you know, in the Genome Project, a public and a private effort. And it actually took the collaboration between them. It really did. That there's a whole story, and the listeners, some of whom know the history there, they'll know exactly what I'm referring to. I think leveraging both investments is, that balance point can be struck. And I think over the years of the project, we'll deal with that, and we'll deal with it in a really thoughtful way.
Thank you, Dr. Kelleher, for coming on the show, explaining this project to us, the Human Proteoform Project. We're excited to hear what happens next.
Erin, thanks so much for the opportunity to share what's going on at Northwestern Proteomics, at Northwestern Medicine, Northwestern as a whole. And, we want to make Chicago a destination for the wild and wacky world of proteomics in the next few years.
Thanks for listening, and be sure to subscribe to this show on Apple Podcasts or wherever you listen to podcasts, and rate and review us. Also, for medical professionals, this episode of Breakthroughs is available for CME credit. Go to our website, feinberg.northwestern.edu, and search CME.